DNA replication occurs in the S phase of the cell cycle, and as any other complex process in our organisms, it requires different enzymes to proceed. Today, we will take a look at the enzymes that play a key role in DNA replication and what they do. The first important enzyme in the process of DNA replication is topoisomerase. As you might know, DNA in the cells is highly condensed. This allows the DNA to take less space in the cell, but it also hinders the replication process. So the role of topoisomerase is to unwind the DNA by breaking the chain and thus allowing the molecule to untwist and then reconnecting it back. The enzyme that goes behind topoisomerase is called helicase. It breaks hydrogen bonds between complementary bases, which leads to the separation of DNA into two separate strands, the leading and the lagging strand. But more about them later. The area that gets formed by the helicase is called replication fork, and that's where the whole process takes place. Of course, we first need a starting point, so another enzyme called Primase synthesizes a short RNA sequence called RNA primer that serves as a starting point. Then, DNA polymerase III binds to the primer and begins synthesizing a new complementary strand by adding nucleotides to the 3' end of the strand that was initiated by the primer. So, we can say that the synthesis of a new strand in the process of DNA replication occurs in the direction 5' to 3'. But as mentioned in my previous video about the structure of DNA, two strands of DNA are anti-parallel, meaning that their nucleotides are oriented in the opposite directions. This not only means that the synthesis of the complementary strands will occur in the opposite directions, but also that the process of replication will be completely different. On the leading strand, the polymerase moves into the same direction as the replication fork, which allows a continuous synthesis of a new strand. On the lagging strand, however, polymerase moves into the opposite direction, but as helicase separates more nucleotides of the DNA, they also need to be replicated on the lagging strand. That is why on the lagging strand, DNA can only be replicated in small portions called Okazaki fragments. Each Okazaki fragment has its own RNA primer, but we need a DNA strand as an end product. So DNA polymerase I replaces the primer's RNA nucleotides with DNA nucleotides. After that, another enzyme called ligase will reconnect the Okazaki fragments together. That is all for today. Thanks for watching, subscribe, and I will see you in the next one.